checking for the final time our technical setup here. I think everything is fine. We will start very much punctual in time so that we do not keep you longer than expected. So for the housekeeping, we have a tight schedule, meaning we will start right now, and you will see a presentation of me, and then we have 10 minutes of discussion afterwards. Of course, that is the 10 minutes where all of you are in the chat. Discussion is only via chat, and then we will allow you, of course, to ask further questions later on. Okay, so thank you very much for joining this. Very new situation for me to talk to you so indirectly and via only electronic interface. Nevertheless, I hope this is quite interesting. It's an interesting time. We have interesting tasks to do in the industry of IVD. So let us go quickly through the topics today. Again, there is more than we don't know than we know. So therefore, if you go home with a lot of questions or leave this presentation, then this is due to the matter and the situation in the moment. So this is what we would like to go through today. It's working. Just a second. Here we go. We have different topics to answer today. Which targets do we see in the determination of antibodies against the virus, and some elevation, some exemplary evaluation of screening data. We will see an overview of available antibody test systems in the market or that are already existing, and what is the difference behind them, and considerations concerning preciseness and what kind of theoretical and experience-based reflection do we have to do according specificity and sensitivity of these new test systems. What is the future targets, target combinations, and what is the way to come to a reliable result in measuring autoantibody or antibodies against the virus in the current situation. So to give you a short, very brief background about INVENT, our mission is the targeted procurement of human biomaterials. That is the reason why we think that it may be worthwhile that we let you know about our opinion on the matter. We always think that precise assays need precise samples, and the current situation is giving us a lot of tasks because our usual way forward is very much in question because we're facing a very new disease, a new situation, and everything is very complicated for us to understand where we are. The INVENT, of course, is certified to two quality norms. You know that we have a donation center on our own here in Hengstdorf. We manufacture controls and calibrators. We have an INVENT clinical services for IVD. These guys are very busy in the moment because clinical validation is critical. We have an R&D unit to develop test systems, especially to do something like proof of principles in test development. But main thing, and that is what we are doing, we take care about human biomaterials. That is basically what we are doing, and we can produce controls and calibrators for that. And we are a little bit above 50 people as full-time equivalents. What do we do? If you need human biomaterials, samples are with us already, yes, and then we ship them to you, or they aren't here, and then we start a targeted procurement project to get to the right samples at the right time in the right volume towards the place of use and need, and then ship them. So our procedures is we can procure any kind of human biomaterial, large volumes, pools, normal disease state, clinical defined samples, cohorts, panels, and we can produce these controls and proficiency tests. So that is everything about INVENT for today. Now let's go into the matter. Everybody is familiar with the acute situation of SARS CoV-2 infection, and this is what we are talking about. The outbreak in 1920, maybe a little bit earlier, we do not know yet, has a big difference how long this virus may already have been circulating, to see what kind of samples you can get and you can get a hand of. As we will see later on, the longitudinal observation of samples is of critical importance at that point. It is a positive single-strand RNA virus, and it belongs, of course, to the family of coronaviruses. So what we have here, and you see this picture, there are mainly two important proteins to be followed: the spike protein that you will know, the nuclear capsid protein, and those two are the ones that are important in characterizing the virus spike and nuclear capsid. And these are very important for two things. The spike protein, as you will see, is the one that is enabling the virus to enter into the cell, and the nuclear capsid protein is the one that is expressing the highest. So in terms of diagnostic values, they are completely different targets towards the answer that we would like to see from a diagnostic toolkit. So the infection cycle, and I will cut that very, very briefly because we have to discuss a lot of other things. You see, the main topic is this one here. The spike protein and the furin cleaving site will be cleaved so that the spike protein complex is divided in S1 and S2, and then via the two receptors, TMPR SS2 and the ACE2, and mainly the ACE2 receptor, the virus has the chance to enter the cell, and that is of importance. So therefore we can clearly see that if we measure antibodies against the virus, those viruses who are directed against the spike protein, and especially the one that is coupling to the ACE2 receptor, is the one that should be neutralizing antibodies and helping therefore to let us know whether or not a patient is protected against the next infection or a recurrent infection. Of course, this belongs to the class of highly pathogenic zoonotic viruses. SARS CoV, the old one from 2002, not existing anymore, and MERS-CoV is also quite of importance, but also is not existing anymore. So I let you know this because there is a huge question all the time towards INVENT to provide in terms of sensitivity and specificity to determine that, especially sensitivity to differentiate between SARS-CoV-1 and MERS-CoV. This is of no real clinical day-to-day importance because those viruses are vanished from the surface of the earth. We don't see them at the moment. That will be very interesting again if there's a re-outbreak of those viruses. It's not the case in the moment. Pretty much more interesting are the classical, the common coronaviruses. The low pathogenic ones that you know, like something like common cold and small respiratory problems. There we have infection rates in the average population of 40 to 80%. And so therefore those are the ones every antibody test shall differentiate or look at and see whether in this kind of environment the specificity is sufficient, so to differentiate from those pre-infected material. And that is quite important, and it's completely unclear until today whether or not past infections or current infections with this low pathogenic common coronaviruses will influence the measurement of the antibodies against SARS-CoV-2. So this is important, and as you will see in a minute how complex the situation is. And here you see that the comparison between, in terms of the nuclear capsid protein, between SARS-CoV-2, so the actual virus, and SARS-CoV, the old one from 2002, and we have a sequence identity of 90% and a sequence similarity calculated of 97.2%. So this is quite a bit of a task for an assay development. Of course, not for an antibody. That is possible for an antibody to differentiate, but you have to look for the right antigen so that you don't have any cross-reactivity. So, and the same like this, of course, holds for the spike protein. You see the two subunits in SARS-CoV-2 and SARS-CoV, the old one. And also you have, again, it's just for the one typo, a huge sequence identity in both of the subunits, and therefore, and those are the one their conformity is this conformational epitope is important to enter into the virus, and this is also the part for the neutralizing antibodies where they are bind to. So basically, if we would like to know, and we come back to this later on, whether or not a patient is protected, the final task should be actually to measure neutralizing antibodies. But this you can only do with a cell culture and very complex under microscope and so on and so forth. So it's not yet automated. But those are the ones who are really giving you an idea about protection from the immune system. Additionally, the spike protein is heavily glycosylated, as you see here. So the light blue one is the spike protein, and then you see all the glycans attached to the spike protein, and you see it's almost completely covered. So therefore, the question is, where are the most important epitopes to be detected by an antibody screening, or if you measure antibodies in a test system, in ELISA or other based test system, automated test system. And therefore, of course, there are a lot of works now going on, either on protein level to find the right epitope, classical protein mapping with 15 MERs and see for the overlap. And the same is already going on also here in this area of Berlin-Brandenburg with the glycan. So there are glycan chips where you measure, where the people are investigating at the moment, it's the Max Planck Institute, which epitope is determined by the glycan structure. Very interesting, and we will see, I'm quite sure that we will see second generation test systems in the future on the market taking care about that. So you see the number are quite impressing. We have 22 glycosylations promoter, and at least 17 of them are all the time occupied, so that's highly glycosylated. Okay, what kind of antigens are available on the market? Usually manufacturer is buying this antigen, is producing by itself, or buying that, or if he's producing by himself, it's all being sold on the market. From there, of course, you have the nuclear capsid protein, which is inside the virion, but upon the spike protein by itself, you see a full length S1 subunit, S1 only the N-terminal domain. You see the receptor binding domain. You can buy the full length S2 subunit and the ectodomain. So a lot of variations in that. Question is which one is clinically relevant. The envelope protein, which is not yet on the market as a commercial test system to measure antibodies against it, and likewise the membrane protein, and also the lots of those non-structural proteins which do not play any role as of today in diagnostics yet. And if you just look at this as you would do if you would like to develop some kind of a test system, and that you see that if you search for them, you get hundreds of hits. Here are 2 different platforms only giving you different qualities of that kind of antigen. And so therefore it is quite important to understand exactly if you buy a test system, which kind of antigen has been coupled to the solid phase. And as you will see, how are antibodies developing? The seroconversion and the immune status. Obviously, SARS CoV-2 causes a completely different, or a significantly different effect, as we would know from our books, as we've learned it at university. So there's almost no difference in time of the onset of IgM or IgG antibodies, and IgA as well. It's not shown here on the picture, but they're almost at the same time. You see the increase in titer of the 2 kinds of antibodies. And this is, of course, 2 or 3 weeks after the onset of disease. And after the onset of disease, before you get symptoms, the PCR goes up, and then PCR in concentration measured in the throat goes down again. Then the symptoms occur, and then the antibody answer, the immune system answer is occurring. What we do not know yet, and that is very, very interesting. We do not know yet. Are those antibodies neutralizing antibodies? It's not been investigated thoroughly yet. How long will they last? So if you see as a comparison, SARS CoV-1 and MERS, is clear that after zero conversion and the first entry of those antibodies, they have a long protective capability. The antibodies built, they are capable or detectable over months or years. Even classical immunization with a vaccine takes 10 years, like tetanus and other diseases. It is not the case here. Obviously, the titer goes down. So it is quite complicated to understand in a longitudinal manner what is what is telling us if somebody has a titer of antibodies, high titer, low titer, medium titer. Avidity is very important, of course, and the target of the antibody. And that has not been differentiated at the moment very precisely. So is it definitely a neutralizing, a protective antibody, or is it just a binding antibody towards the virus? The latter one is actually, in most of the cases, in normal viruses, I must say, is sufficient. So you get an infection with the virus and you see from any kind of an antibody against this virus that the person has immunity because the person has seen the virus, the immune system has reacted, so it's absolutely plausible to say this person is immunized and protected. Here now, it is not clear whether this is the case, and also it is not clear whether or not this bunch of antibodies that are being produced against the virus do not have any severe negative side effects. Yeah? So first cases are reported of MS-like or Parkinson-like or Guillain-Barré-like syndromes that are established in patients as a side effect of SARS-CoV-2 infection. So that is of high importance if we start to find a vaccine producing antibodies, and if those antibodies, maybe the bad ones, are then having side effects that lead to a long-term destruction of the myelin coverage of neurons, then we have a huge problem while trying to do something good. Okay, back to diagnostics. These are the test systems you can see on the market at the moment, and it's only an excerpt. So as you see, the list goes longer. Here, the three dots telling you this list goes on, and it's been updated on almost on a daily basis. And what I would like to point out is you see the certification behind those in the last column here, certification. So either it's in Europe, CE, IVD, or it's FDA, and there's something that is new. A lot of those tests are only being allowed on the market in the US according to emergency use authorization. So it's compared to what we are used to in terms of quality standards. This is definitely a lower standard. So we have to take care, and will be a lot of work to be done in the future to validate those test systems. And you see here clearly that the antigens are, all of them are on the market, and you can measure them, and which one of those is the clinical, the right one, and which kind of clinical results we shall derive from measuring one or the other. It is not clear yet. So giving you these numbers in a different perspective, there are more than 300 regulatory authorized diagnostic test kits on the market. Of those are 160 serological tests. With authorization, in total 230, so the rest of them without authorization. And if you look at them, then you see the distribution towards CE, FDA emergency use, and FDA do not distribute, so different classes of FDA regulation applying. The 31 authorized serological assays that we know, you see they are measuring different classes of antibodies, IgG, IgG, and IgM, or total IgG. So different scope of the test system, and then you see also the distribution towards spike protein, nuclear capsid protein, or a combination, which may be very, very useful also clinical in the future, combination of capsid and spike protein. We will see which one is in the end the most useful, but that has to be evaluated in longitudinal, long-term studies going forward. So some of them, just to show the difference again, we've just picked randomly 4 of them. One generic assay has been produced close to our place here, therefore we're quite happy also to present that here. They are soaring quite quick on the market. Roche, of course, with a lot of effort, and IBL as well, quite quick, and generate assays. And you see from the antigen different strategies toward that, either the holo approach from the lower one, or you go for S1 and S2, or the capsid protein. Again, should usually show just the immunization should work. Here's the technology behind that. So whatever you can know is on the market, you can find easily available now, automated system, non-automated system, manual systems on the market. But then we have the next point. It is like always in measuring antibodies, there is no standardization. We have arbitrary units, we have just the cutoff and then an index towards the cutoff. We have specified units, Novatec units, therefore you know who is producing that. We have a binding index, so there is no standardization. Again, it would have been a chance to move on in this new field and start the standardization from the beginning, but I think that will be a lot of work for the future to harmonize them. Sensitivity is given here. You see a huge variety on the number of samples this sensitivity has been measured, and also the high question is, and we will see that on the next page as well, or the intensive question is days after onset of disease, and then sensitivity is pretty much a function of days after onset of disease. So the antibodies are just developing. Specificity measured against blood donors, we have a comment on that as well, and interfering substances are measured quite nicely, and you see a lot of companies have taken also the cumbersome work to see for interferences with MERS, which to our understanding is not necessary because this virus does not exist anymore. So here you see the what I just mentioned. The sensitivity is increasing in longitudinal measured in a sample in dependence on days after onset of the disease or days after positivity in the PCR value. We can see that here with some data IBL provided nicely, and you see that the sensitivity is going up as a function of days post-syndrome onset. So that is obvious, and that you have to know that because that has of course a huge influence on positive and negative predictive value and the usability of the test system by itself. What did we do? We compare just very quickly just 6 samples where we know the clinic of, and what we can see is that in IgG, for instance, the same tests are measuring the same. Here too, we can continue that. It's not specific for IBL and Roche. We do not want to judge any of those test systems. We just would like to say that some of them are measuring very equally, but you see also differences here where you see positivity and negativity distributed over the test system. That is and is promoted in IgM. IgM is not that specific. It's not a ripened antibody, so therefore this is explainable. But that will be a long-lasting task to see for that. So if you are using this kind of test system, take care. If you change the test system, if you change your test system within a study and the like, or while treating patients, you have to be aware there is no harmonization yet. That there's we are just in the beginning of that. A lot of things are possible. We have to look at this very careful, and we have to see how can we provide tools to do this, to establish the capability to compare different test systems. Here, another thing, we just followed up here 45 samples. Now we are in hundreds and thousands of samples that we followed up to see again different company here, Epitope Diagnostics and IBL, and an in-house ELISA that we built up ourselves just to see how this is all working. And what we can see is we measured the different, the different antibody classes, and with the different antibody classes, IgG, IgM, IgA, we see quite a high positivity rate all over those samples. And that is a little bit in contradiction to what we see in the measurement of the specificity that we had beforehand with the blood donors. And also the question is, how old are the blood donors, and do we have an increase in positivity rate while either the test is technically more sensitive, or when the blood donations are younger, so that people having no symptoms, having had the infection, and then going back again to a regular business of being a blood donor. And there we see, you know, quite a number of people being positive. So none of those samples has been selected to be positive in terms of the disease, or PCR positive, or having symptoms. So they are either different diseases or healthy, self-reported healthy people, and so on. So we see there are a lot of inconsistent results between the samples, and that will keep us busy for the next years to come, I would predict that. So in conclusion, we have quite a diversity of test system, and of course we have quite a diversity of presented antigens from the different manufacturers. The immune response is different from what we know from the other viruses. So obviously IgG and IgM and IgA appearing quite quickly and at the same time. We do not know yet how long the protection of IgG is, so the long-term protection of an infection is totally unclear. Of course, there will be a memory effect, which is clear. We have all we may have to look towards the helper cells, the effector cells, to see for the long-lasting, and memory cells for the long-lasting immunization. But just from the IgG status, this will be tricky to do so. The clinical use, therefore, is quite critical. We cannot say anything yet how useful the test systems are. The clinical development of the disease is also unclear. Side effects, later effects of this demyelinization effects, or heart conditions. ACE2 is quite common in cardiocytes. Does that offer protection? And time, of course, time distribution in the future. Harmonization and standardization is a key, and the predictive value, of course, has a lot of things to do with the prevalence of the test in the moment. The prevalence of people having recovered from the disease is so poor that the predictive value is poor in the moment. Positive, negative predictive values are not at the rate that you can use that already as a diagnostic tool. How can we support you? We have positive controls for characterization of a test system. We call them positive specimen. You can order them with us. Analysis of different antigens and interfering substances. We standardize that in terms of a panel. So you can buy this panel 10 times, 20 times, 100 times, so that if you compare two test systems, or you would like to introduce different test systems, then you're able to compare them on a standardized method. As long as we don't have any international standards, we think that is the way to do. The validation of the test system by determining specificity. We have specificity panels for SARS CoV-2. And large volume donations, of course, if you would like to produce your own controls or bring them on the market, that we can help you. Be aware that positivity is given only in the test system that we measured. So there is not a general positivity to be given in the moment, because there is no standard and there is no measuring system yet at the moment. Please find more of our products that we would like to help. That it is definitely a target for us, with us on these two pages given here, Invent Diagnostically and Central BioHub. And of course, Mrs. Esslinger from Invent will be happy to assist. Thank you very much for your attention. I cut it down the chance to discuss this pretty much. I'm sorry for that, but nevertheless, I hope that the chat is still open, and I hope you find that useful. Please let us know whether you find that useful, because we would be very happy to start this as a series of several of those seminars to come in the future, and going into detail the more we know by ourselves, and let you know, because in the moment it's such a huge variety on information on the market. So there will be plenty of things to do and a lot of work to be done, but we have to do this very precisely and correct as we can do. I thank you very much. If there are any immediate questions that cannot be answered by the team sitting here, thanks to Julia, thanks to Talena, please continue to ask questions. We would be very eager to answer them. If you have different topics that we should address in future seminars to come, I'd be very happy to focus on that as well. That was the first. We hope you find that useful. We by ourselves try to do everything to help any of you to promote your work, your business, and your research as good as we can. Yeah, thank you very much. I think we are pretty much good in time. This is how it should be. Webinars are all forcing us to be precise and punctual. We like that. Thank you very much again, and see you soon.